we will talk about pyruvate dehydrogenase. Pyruvate dehydrogenase is an enzyme which converts pyruvate, which is formed at the end of glycolysis, to acetyl coenzyme A. Simple. And it removes carbon dioxide and it converts NAD plus to NADH. And this NADH, which is formed, will then enter the electron transport chain and it will form ATP. This is a basic overview of what pyruvate dehydrogenase does. But the pyruvate dehydrogenase is actually a very complex structure, which means that it contains 3 apoenzymes and it has 5 coenzymes. So 3 apoenzymes are the inactive proteinaceous part of an enzyme. So the pyruvate dehydrogenase will actually have 3 apoenzymes. So the 3 apoenzymes are E1, E2 and E3. Okay, the pyruvate dehydrogenase enzyme has 3 apoenzymes, E1, E2 and E3. These are the names. Okay, so the E1, it is pyruvate dehydrogenase. The E2 is dihydrolipoamide transacetylase, and 3 it is E3. E3 is dihydrolipoamide dehydrogenase. The name will be understood once you understand the multi-enzyme complex, and also you will understand what apoenzyme has what coenzyme. So let's look at the reaction that is happening here. Okay, so at the end of the glycolysis we know pyruvate is formed. Okay, now this is the pyruvate which is formed at the end of glycolysis. Now what happens? The pyruvate, in the presence of pyruvate dehydrogenase, okay, will split into carbon dioxide and a hydroxyethyl group. Okay, so what is happening? The pyruvate is formed of CO2 and hydroxyethyl group. So what pyruvate dehydrogenase does is that it splits the pyruvate into carbon dioxide and hydroxyethyl. But the pyruvate dehydrogenase is an apoenzyme, so it needs to have a coenzyme. Coenzyme are the substances which activates the apoenzyme. So the coenzyme of pyruvate dehydrogenase is thiamine pyrophosphate, which is vitamin B1. So the coenzyme is pyruvate dehydrogenase and the apoenzyme is pyruvate dehydrogenase and the coenzyme is thiamine pyrophosphate. Okay, you have understood this. I will write it again. Okay, so what happens? We have the pyruvate which is formed at the end of glycolysis, and then it will convert into carbon dioxide and a hydroxyethyl group in the presence of pyruvate dehydrogenase, but it is an apoenzyme, so it needs thiamine pyrophosphate. So I will write here thiamine pyrophosphate. The TPP will activate PDH and it will bind, the TPP will bind with hydroxyethyl group of pyruvate, and hydroxyethyl TPP is formed from this. Okay, this is the first step, and this PDH is the E1. Now we will talk about E2. Okay, the second apoenzyme. What is happening in this reaction? Okay, the second E2, it needs the presence of a lipoamide, oxidized lipoamide. So the oxidized lipoamide along with the E2, okay, is converting the hydroxyethyl TPP into an acetyl lipoamide. What is happening? The TPP is recycling back to this TPP. Okay, this TPP is reused again, and this hydroxyethyl group in the presence of E2 with its coenzyme, which is oxidized lipoamide, is getting converted to acetyl lipoamide, and the name of this E2 is dihydrolipoamide transacetylase. What are we doing? We are transferring the acetyl group to the lipoamide, so it is a transacetyl. Okay, now we have the acetyl lipoamide in the presence of dihydrolipoamide transacetylase enzyme, which is the E2. I will write it down. So we have this hydroxyethyl TPP. So the TPP will go back here, very easy. And now we have E2, which is the dihydrolipoamide transacetylase, but it is an apoenzyme, which means it is inactive. For its activation, it will need, what will it need? It will need oxidized lipoamide, and then what will be formed? Acetyl lipoamide will be formed from that. Now this acetyl lipoamide will again form lipoamide, okay, and then the acetyl group will be removed from this acetyl lipoamide. What is happening? So we have a reduced lipoamide formed from the acetyl lipoamide, and what happens to this acetyl? It will bind with coenzyme A and will convert into acetyl coenzyme A. Where is this acetyl coming from? This acetyl is coming from the acetyl portion of the acetyl lipoamide. So the acetyl comes here and the lipoamide goes here. And in this reaction also E2 is used, which means that the coenzyme of E2 are two. We have oxidized lipoamide number 1 and coenzyme A. Okay, so first we have apoenzyme we talked was PDH, and its coenzyme was TPP, which is the thiamine pyrophosphate. The second coenzyme of the PDH complex, it is dihydrolipoamide transacetylase. And what are the coenzymes? This and this, which means we have a lipoamide and coenzyme A. Okay, so we have talked about two things. Now this reduced lipoamide will get converted to oxidized lipoamide by how? By converting this FAD to FADH2, and this FADH2 will again form FAD by converting the NAD plus to NADH. Okay, so what it is using? It is using FAD and it is using NAD plus in the presence of a third enzyme E3, and this E3 is also a dihydrolipoamide, but it is a dehydrogenase. Any reaction involving NAD plus, the enzyme would be dehydrogenase. So here also we have dihydrolipoamide dehydrogenase. Let me write it down. So this reduced lipoamide is converted to oxidized lipoamide in the presence of an enzyme called dihydrolipoamide dehydrogenase, because it is consuming an FAD and converting it to the FADH2, and this is converting back to the FAD by converting the NAD plus to the NADH. Okay, so this is the third apoenzyme. So the third enzyme is dihydrolipoamide dehydrogenase. And what are the coenzymes for it? So we can clearly see that the FAD and NAD plus are the coenzymes for this, right? So we will write down FAD and NAD plus. So in this table also you can see that the pyruvate dehydrogenase enzyme and the coenzyme is TPP, which is vitamin B1. Next we have the dihydrolipoamide transacetylase, which has a coenzyme lipoamide and coenzyme A, very clear, and dihydrolipoamide dehydrogenase with the coenzyme FAD and NAD plus. That's it about this video.